Hello, in this video we will discuss about the death after vaccination and what is the conspiracy theory. Conspiracy theory this is because the after vaccination it will not die. People will not die because there are many reasons, and this is the myth basically. For example, here I am explaining the conspiracy theory is an explanation for an event or situation that invokes a conspiracy, sinister and powerful groups of often political in motivation when other explanations are more probable. So in this way, the other is it can be a jealousy or something which that is developed the theory for inhibition of that things which that is progressing. So that is the inhibition mechanism for production of theory for the fearing to the community. So this is the basically controlling of the world, and is the vaccination business flop idea, strong health inhibit in different country and human race as well as competition, because there are many countries which that is developing and growing due to the vaccination, and vaccination provide throughout the world, and there are many things which that is related to the vaccination. So in this way, the death after vaccination theoretical explanation will develop. So in this way, this is basically, first of all, we should look at the mechanism of the action of the vaccine. So here is after the death, a death after vaccination is impossible. How it is true, and let's begin to understand. This is a person which that is healthy and living before 2 years after vaccination. So we are putting the vaccine into the blood circulatory system of this person. So in this way, basically is the vaccine is basically contain inactive or part of the virus or RNA virus, RNA vaccine. So the RNA vaccine is coated by the lipid monoparticle, nanoparticle. So the lipid nanoparticle which contain RNA. So this is basically the cell. Look at this. This is the cell. After the vaccination, the RNA will enter into the cell, and after the entry into the cell, go into the cytoplasm where the endoplasmic. This endoplasmic reticulum is used for the synthesis of protein by the using of the viral RNA. So after the synthesis to protein to show through our Dr. manner B antigen presenting cell, antigen presenting receptor, which that is the MHC class 1 and 2. So after that representation of the spike protein from the synthesis into the cytoplasm through the machinery of the host cell, for example the human. So manner B is also produced due to the when go into the nucleus to bind the DNA with the nucleus, so that produce the another spike protein will attach in the upper part of the cell as a receptor like structure to show that this is virus like. So the endoplasmic reticulum is here, which that is I am drawing the bigger picture. So the rough endoplasmic reticulum is the rough endoplasmic reticulum. So basically the RNA is converted into the reverse transcription by the reverse transcriptase enzyme, and after the reverse transcriptase enzyme, basically it is impossible but it can be bind. You can suppose it, and basically this will be bind, and after the binding it will be transcribed and the human cell nucleus DNA binding. So this is the recombinant DNA will be occur, but eventually this is impossible, we will also understand. So the messenger RNA will produce after the transcription, and this messenger RNA will be bind with the rough endoplasmic reticulum containing ribosome, and after the binding with the ribosome, the smaller subunit and bigger subunit bind with the messenger RNA to synthesize through transfer RNA to synthesize protein. This synthesis of protein mechanism is known as the translation and produce the polypeptide. This polypeptide will further mature into the endoplasmic reticulum, as well as after the maturation the endosome formation, which that is bind with the Golgi bodies for further mutation, further maturation, and cis phase to trans phase binding with the Golgi bodies to become a spike protein endosome formation, and that vesicle which that contain is basically spike protein. And this spike protein is basically is used for the representation for the immune cell for activation of that things. But in the case of DNA as a genetic material binding to show the spike protein with the whole cell, and it is possible, but eventually it is not possible because the apoptosis is very important. During the apoptosis, the cell CD8 cell will be killed that infected cell because the recombinant cell will not live longer and apoptosis will be occur. So here is the DNA interference if we will discuss about. So the proofreading will be occur. In this way the proofreading will check the DNA. After the checking of the DNA, if foreign DNA are present, so the apoptosis can be occurred because the recombinant cell will be apoptosed, the cell death, necrotic cell death, so the necrotic apoptosis. But while on the other hand here, if the on the case of the infection or death, so it can be natural killer cell, macrophage, mast cell, and dendritic cell which can be kill this cell due to the spike protein representation. So in this way here is you can see, otherwise no apoptosis if, so the natural killer cell, macrophage, and dendritic cell will kill the whole body. So that is why the apoptosis will be occur, and it is possible because the immune system will detect continuously with the infected cell or changes cell, the cell changed. So the again infection will be caused after the vaccination. So the infection will again cause to when entry of the virus, so the enter into the tissue. After entering into the tissue to become an infected cell, so it is also basically is also already the spike protein are present. For example, if present, so it will not immune response will be occur. So the MHC class 1 and MHC class 2 molecule will be represent like this. So in this way the CD8 cell will be kill this antigen containing cell, infected cell, to become apoptosis through CD8 cytotoxic T cell. So in this way, when virus will be entered, so the immune system is already activate and bind with the opsonization with the antibodies through B memory cell. So the important thing is that you should need to understand that the immune cell will clear the infection, clear the problem. So already that is why the allergic reaction will be occur after 2 days, 3 days, but not occur in after 2 year or 3 year, or death will not occur because here is you can see this the T helper 1 cell, CD4, and CD8 T helper 2 cell, which that will be bind, and after the binding to show the MSC class 1 molecule to naive B cell. Naive B cell will be activate into the plasma cell and B memory cell. After the plasma cell and B memory cell will opsonize that antigen containing tissue, so it will be kill that tissue by the opsonization. So the killing of this tissue is very important and crucial for play important role. So during the 1 cell or 2 cell recombinant will be apoptose, or B memory cell and plasma cell will kill that, opsonize that, and not dividable. So the mitosis will not occur. So that is why the death is impossible. So in the case of here is you can see this is the basically the virus, and after the detection by toll-like receptor with the macrophage and dendritic cell to become an endocytosis. After the endocytosis, it will be represented by MHC class 1 molecule. After the MHC class 1 molecule, here you can see this is MHC class 1 molecule detection to become endocytosed. After the endocytosis, so the Golgi body, endocytosis will also trigger the opsonization through a B memory cell recruitment. So here the naive T cell will proliferate into the Th1 T helper cell 1 and T helper cell 2 by the recognition by MSC class 1 molecule through dendritic cell. So in this way, important things is that you should need to understand that allergic reaction are there many complication will be occur after 2 or 3 days or 1 days. So that is why that reaction is due to the reaction due to the synthesis of the spike protein and the immune response, and inflammation will be occur. And allergic reaction, apoptosis, tissue damage, and blood clotting allergic reaction. But after 5 to 6 days, because antibodies production but not possible to take more time, because before the 5 to 3 days antibodies in us not producing, so the inflammation will increase, but the rapidly produced antibodies will clear the infection and live longer and not will be die. So that is important things are death after vaccination is impossible, and that is the conspiracy theory about the death after vaccination. So thanks for watching. Bye.